everyone, welcome to your Neet English batch. Good afternoon, how are you doing? Sorry, I think there was like a 2-minute delay. Okay, Anam, good evening, good afternoon, Sinchana, good afternoon. Akshara, Pavan, Sahiba, Sahiba, Nancy, Sushmita, Yogita, everybody else, good afternoon. Is the audio and video clear? Okay, so in the classes, other batches that I've been taking, in some batches, like in 2 batches, yesterday they complained that they're hearing some background noise. Do you also hear it? Like right now as I'm talking, is there some sort of a fan sound that you can hear in the background as a noise? Was that there yesterday? Is there any noise at all? No noise? Okay, fine. All right. So I got it fixed. There was some issue with the audio filter, but I just wanted to confirm that there is no noise. I'm doing great. Thank you for everybody asking. And yesterday I had circulated a form in the group, a Google form in the group, where I had asked you all to fill the forms. I got a lot of responses on the form and I got a lot of responses personally. You've been sending me messages. I read a couple of them and I replied to a couple of them. But if I have not replied to your message, don't feel bad. It was like too much of a flood of messages that I got. So I may have missed 1 or 2, but after class again I will sit down and go through it, and if I have missed replying to your message, I will reply. The best thing for you to do is you can type it, you can fill the Google form instead of sending me a personal message, because there I will have a track of everything. On my Telegram, it's possible that 1 or 2 messages can get, I can miss them, but over there that possibility is not there. Also, if you're worried about will anybody see it, will they find out who you are, what your story is, you don't have to worry about it. I have not made any field in the form mandatory. You can fill the form even without writing your name, without giving your phone number. You can fill the form, okay? So it's going to be completely anonymous. I will not judge you even if you tell me your name. Apart from me, nobody else has access to that form. So you can be sure of that, okay? You don't have to worry about it. So, yeah, that would be better. If you fill the Google form, good. I'm talking about our Telegram group. Okay. So, did everybody read after yesterday's class? Did you go through the experiments and learn? Are you perfectly thorough with all of that? Does anybody have any doubts from yesterday's class? Brilliant. I will keep the forms open till Wednesday evening. After that, I will sit and go through it and then do my homework, and then we'll have our session. Okay? Today after class, I will ask the form to be posted in the community section as well. Yes, Pramod, you have to send it to me. You have to fill that form and send it. Just give me 1 minute. For some reason, this thing is not opening. Just 1 minute. All right. So, okay, Rahul doesn't know anything about the form. Some of you have questions about the form. Yes, Pratik, you can describe it in detail in the form itself. For whoever is facing trouble in studying every day, you are distracted a lot, or whatever could be your issue that you're facing that's keeping you away from your studies, you can talk about it in the form. All right. So in the form, you will have to enter details like your name, number, are you a fresher, are you a dropper, whatever it is, and what are your major distractions. If you have to share anything specific, you have provision for all of that. But none of the fields are mandatory. If you choose not to let me know your name, that's fine. You cannot fill that. If you don't want me to, if you don't want to give me your phone number, that's fine. You cannot give that. Whatever you want to convey, it is possible for you to convey it through that form. I'm going to analyze that form, and according to which is the most pressing issue right now that most children are facing, I'm going to conduct sessions that will help you overcome those distractions, overcome those issues. Now, where is this form? Where can you find this form? There is a group on Telegram called Yaki Neet English 2025. Okay, so in that Telegram group, I have posted this. If you're not a part of this group already, I think the QR code must be somewhere in the community section. You can scan that and join it. Otherwise, I will see if I can post the link of this Google form on the community section today after class. Alright, you still have time. If you have not done it yet, it's okay. Till tomorrow evening, I will keep it open. So as and when you can fill in those forms. And by the end of this week, I will schedule a Zoom session where you all can interact, like not just type. You can turn on your cameras, turn on your mics and talk and communicate, and we can have that session. Alright. How to get the form, Shriya? Just give me some time. I will figure out a way. I will let you know how to join the Telegram group. On the Telegram group, I will post that link again today, and over there you can access, or I will post it in PW Yakin Batches community section itself. Now, let's start with the session. Okay? I think I've answered most questions about the form and everything. So we will start with the session. Everybody give me a thumbs up. Nikita, why do you need Akshara's Telegram ID? I'm telling you that I'm only going to give it to you. You don't trust me. Cool. I will disable the comments for a bit. Okay. What did we do yesterday? Fine. So in the last class, we spoke about some early experiments and what we already know about photosynthesis. We know the final equation of photosynthesis. Photosynthesis requires carbon dioxide, water, chlorophyll, sunlight, and as the products, it will give us carbohydrates like glucose and oxygen as a byproduct that we all inhale. So basically, we're dependent on plants to survive. If not for them, we will not get our food. If not for them, we will not get our oxygen, right? This is basically the overview of what was taught yesterday. And in the end, I gave you a brief introduction to photosynthesis. Let's recap that once quickly. So yesterday, I said that photosynthesis does not happen in a single step. Photosynthesis happens in two phases. Photosynthesis happens in two phases. The first phase is called the light reaction. It has many other names. It can be called light dependent reaction or photochemical phase. And the other one is called dark reaction, or you can call it light independent phase or biosynthetic phase. What basically happens during light reaction? Sunlight is trapped, right? So you have sunlight, the energy in the sun. If the sun's energy has to be trapped, then obviously who has the ability to trap that energy? Pigments have the ability to trap that energy. Pigments like chlorophyll will absorb that energy present in the sun. Right? You have pigments like chlorophyll that will absorb this energy. It will absorb this energy, and what does it do? Does it immediately synthesize glucose and synthesize glucose by utilizing carbon dioxide? No, it doesn't. It will absorb this energy and convert it into a form that plants can use later. What are those forms? What are those energy-rich compounds? They are your ATP and NADPH. So that will synthesize energy rich molecules ATP and NADPH. In this light reaction itself, water molecule gets used and oxygen gets liberated. Alright, now what happens in the dark reaction? In the dark reaction, you can see that carbon dioxide gets reduced to form glucose. For this energy is required, and this energy comes from ATP and NADPH. Okay, so all of this is happening in the chloroplast. Where exactly in the chloroplast, we'll understand. Okay, so first, I don't think I mentioned the full form of ATP and NADPH yesterday. ATP stands for adenosine triphosphate. NADPH stands for nicotinamide adenine dinucleotide phosphate. Okay. N nicotinamide, A adenine, D dinucleotide, P phosphate. What is H? H basically refers to the reduced form of NADP. When NADP is reduced, we say it's NADPH. Okay. So H does not stand for anything here. ATP is adenosine triphosphate. Alright. So we know about this already. I hope there is no doubt in this. Today, I want to start by explaining where exactly photosynthesis takes place. Where does the light reaction happen? Where does the dark reaction happen? So photosynthesis generally happens in the leaves, right? In some young stems also, if the stem is green, photosynthesis can happen. But if you take a leaf, take a stem, and over here, let's just focus on one leaf. Do all parts of the leaf have chloroplast? Let's take one section of the leaf and look at it. In a leaf, we've already studied an anatomy of flowering plants. You have your upper epidermis. Of course, some stomata may be present there. And you have your lower epidermis. In the center, wherever veins are present, over there, you can find your vascular bundles. For me, right now, this is not of importance because I'm talking about photosynthesis. Where does photosynthesis majorly occur? In the mesophyll cells. Over here, you have many cells called mesophyll cells that have abundant chloroplast. Right? There's a lot of chloroplast present in these mesophyll cells. I've taken a dicot leaf, so you will have palisade and spongy parenchyma. Now, let me just focus on one of these cells. I'm going to focus on one mesophyll cell here. Okay. These are your mesophyll cells. What is one mesophyll cell going to look like? Like normally, what all parts plant cells will have? This mesophyll cell also will have the same parts. Plant cells have an outer cell wall, right? It has a cell wall. Inside the cell wall, you have the cell membrane. Within the cell membrane, you have cytoplasm Within the cytoplasm, you have a lot of organelles. But what is of interest to us right now? What is interesting to us right now with respect to photosynthesis is one type of plastid called chloroplast, where photosynthesis will take place. So within this, you can find chloroplast present. Chloroplast will be present. What is the structure of chloroplast like? Do you remember? We learnt about it in Cell, the Unit of Life chapter. So what does a chloroplast look like? A chloroplast is bound by two membranes. This is a section of the chloroplast. It has an outer membrane which is more permeable, and it has an inner membrane that is comparatively less permeable. Right? Within that, what do we find? We find a fluid matrix that is called the stroma. What do you find inside the stroma? Inside the stroma, you can find membranous structures. And these membranous structures are found in the form of sacs. For example, this is one Taylor Angdi slip. Okay. Your thylakoids kind of look like this. Okay, they are flat sacs. It has a membrane. It is membranous. Inside the sac, you have space. This space is filled with a fluid similar to cytoplasm or similar to stroma, you can say. It's not empty, khali kind. It has a fluid within it. This is closed on all sides. Okay, so this is what we call a thylakoid. Within the chloroplast, you can find several such thylakoids stacked one on top of another. Each one here is a thylakoid. This entire stack is called a granum. You have many such grana present inside the chloroplast. Okay, I'm reminding you, you've already studied this, it's not new. You've learnt about it in Cell, the Unit of Life. This is like a reminder. I'm not talking about the other not important things. Of course, inside the stroma, you will find 70S ribosomes, you will find circular DNA, but that's not important to us right now. Right now, what is important, that's the only thing I'm focusing on. Alright, so over here, you have several grana. What I'm going to do right now is consider just one thylakoid. Let's take a look at just one thylakoid. What is this thylakoid going to look like? I just showed you, it looks like a sac. It looks like this sac. Right? This is a thylakoid. There is space inside the thylakoid. What do you find on one side of the thylakoid membrane? If this is your thylakoid, what do you find? This is the thylakoid membrane. It's covered like this. What do you find? Oh, why won't you cooperate, paper? Okay. So, if this is the thylakoid membrane that's completely covered, this is the membrane of the thylakoid. On one side of the membrane, what do you find? You find space. We call this space the lumen. Look at this membrane alone, this top one. On one side, you have the lumen. What do you have on the other side of it? Take the membrane, one side. Towards the inside, the lumen is present. What's present on the other side? On the other side, you have stroma. Because where is this present? That's present here. If it's present here, on the other side of the membrane, what will it have? Inside it has the lumen. On the other side, it should have the stroma. So in this thylakoid membrane, towards the inside, you have the lumen. Towards the outside, you have the stroma. What is this thylakoid membrane made up of? Let's understand that. If you remember, when we spoke about the structure of plasma membrane, we called it the fluid mosaic model. Do you remember that? When I told you about the structure of the plasma membrane or structure of the cell membrane, I mentioned that everywhere there is a membrane inside the cell. it's going to have the exact same structure. The plasma membrane, membranes of the ER, Golgi, lysosome, nucleus, chloroplast, mitochondria, every membranous structure will have the same chemical composition like phospholipids and proteins and all of that. Okay. So here, this is a membranous structure. This thylakoid is a membranous structure. So in the membrane of the thylakoid, you will find the same things. You will find phospholipids being present. You will have phospholipids forming a bilayer. Okay. Similar to how you found proteins being present in between the phospholipid bilayer in the plasma membrane, you will have something called pigment complexes here. Okay, so within the membrane of the thylakoid, you will have what is called pigment complexes. What is this pigment complex? A pigment complex is a group of pigments. Okay, so over here, generally what students think is when we say chlorophyll is present in the chloroplast, they assume that chloroplast is dissolved in the stroma. But chlorophyll is not present in the stroma. It is present in the membranes of the thylakoid. It's present in the membrane of thylakoid. It's also present in the stroma lamellae. Okay, they're present here and they're also present here in these membranes. Okay, I want you to remember the names of this. We will be using it very frequently. These are the grana lamellae. Whatever membranes are there in the grana, they are called grana lamellae. Whatever membranous structures are connecting the grana, they are called your stroma lamellae. So, in the membranes of both grana lamellae and stroma lamellae, you will find pigments present. Pigments are not present singly, alone, but they are present in groups of hundreds. Hundreds of pigments along with some proteins and electron carriers will form a complex, and that complex is called a pigment complex. For now, just let's call it pigment complex. Within this pigment complex, you can find hundreds of pigments. So, if light reaction has to take place, light reaction requires pigment chlorophyll primarily. If chlorophyll is present in the thylakoid membrane, where do you think light reaction will take place? Light reaction obviously has to take place in the thylakoid membrane itself, right? So, because the primary requirement for light reaction, chlorophyll, is located here. Therefore, light reaction will take place right here in the thylakoid membranes. Light reaction happens in thylakoid membranes. Now, what is the product of light reaction? At the end of light reaction, what do we get? Right now, we are saying that light reaction is taking place here in the thylakoid membrane. At the end of light reaction, ATP, NADPH, high energy containing molecules are produced. Now, where is this ATP and NADPH released? Once they are produced, there are two possibilities. They can be released towards the lumen or they could be released towards the stroma. Where dark reaction happens depends on where this is released. So, the products of light reaction are released into the stroma. ATP as well as NADPH, that is released into the stroma. For dark reaction to take place, whatever enzymes are required, whatever energy rich molecules are required, all of that will be present in the stroma. So the dark reaction will happen in the stroma of the chloroplast. Though photosynthesis we say is happening in the chloroplast, light reaction and dark reaction do not happen at the same place. They happen at different locations depending upon the requirements. For light reaction, what do we need? We need pigments. Where is the pigment present? In the thylakoid membrane. So light reaction happens in the thylakoid membrane. For dark reaction, what do we need? We need certain enzymes. We need ATP, we need NADPH. Where is that present? That is present, located in the stroma of the chloroplast. So our dark reaction occurs in the stroma of chloroplast. Is that clear? Is this clear so far? Does anybody have any doubts? Yes, light reaction can occur in the stroma lamellae also. I told you no, Farshida. Wherever pigment is present, over there light reaction will happen. It can happen in the grana lamellae or it can happen in the stroma lamellae also. Okay, so if it's clear, if there's no doubt, I will move on. Last part. Okay. Last part I assume is this. So the thylakoid membrane will have a structure, phospholipid bilayer, similar to our plasma membrane. In plasma membrane, you have phospholipid bilayer and then you have proteins embedded within the membrane. Even over here, you will have a phospholipid bilayer, but instead of proteins of course will be present. Apart from proteins or along with proteins, you will also have certain pigment complexes. In each of these pigment complexes, you will find hundreds of pigments. Okay, so because pigments are here, light reaction will happen in the membranes. Because the products of light reaction are released into the stroma, the enzymes required for a dark reaction are present in the stroma. Dark reaction will happen in the stroma of chloroplast. Alright, if this is clear, I will move on. I will disable the chat for some time. Okay, now I told you that there are hundreds of pigments present in the pigment complex, right? Are all hundreds of pigments over here chlorophyll? Is chlorophyll the only pigment present in leaves? Is it the only pigment that is responsible for photosynthesis? Are there any pigments at all involved? So let's try to answer that. You may have seen during different seasons and in different plants, leaves will change in color. Sometimes the leaves that have appeared green for most of its life will start appearing yellow or orange or some other color. Now, understand the difference between variegated leaf and what I'm saying right now. In variegated leaf, every leaf in the plant from the time it is formed, it will have more than one color. That's a different story, what we learnt about yesterday. What I'm talking about here is, you take any plant, during certain seasons, it will change color. During spring, it will change color. What was entirely green may start appearing entirely yellow or entirely orange or somewhat like rust orange. So it will change color. What does that mean? What basically it means is that apart from chlorophyll pigment that will give green color to the leaf, the green will also have other pigments that are responsible for other colors like yellow, orange and all of that, brownish orange and all of that. Because chlorophyll A is so abundant, the leaf is appearing green to our eyes. What happens when leaves grow old or in some plants that shed leaves during spring? Chlorophyll will break down. Once chlorophyll breaks down, you cannot see green color. At that point, other colors will start appearing. Does that mean these other color producing pigments formed at that time? No, they were present, but we could not see it because chlorophyll's abundance was masking its effect. Right now that there is no chlorophyll, you can see the effect or the appearance of different colors because of those pigments. Okay, so in these pigment complexes, you will have chlorophyll for sure, but along with chlorophyll, you will have other pigments also. If you remember when we studied about plant kingdom, we learnt under algae that there are 3 groups of algae based on pigments. We have classified them into red algae, green algae, brown algae. All of them have chlorophyll A. If you take red algae, it has chlorophyll A, it has another type of chlorophyll called chlorophyll D, and it also has phycoerythrin, right? Phycoerythrin is also present along with chlorophyll A. Then if you take brown algae, there also you will have its own pigments. If you take cyanobacteria, blue-green algae, there it will have its own pigments. What all photosynthetic organisms have in common is chlorophyll A. But along with chlorophyll A, you will have other pigments as well. Okay, so how does one find out what are the different pigments that are present in a leaf? What do? We do something called paper chromatography. It is, I think in your 12th grade, 11th grade, I don't know when, you have this demonstration experiment in your practicals which your teacher might demonstrate to you. If it's not there, it's okay, I will explain it to you anyway. So what we do over here is you can take any leaf, palak, spinach, both are the same, or some random leaf that you can find. Take it and grind it in a pestle and mortar. What are you going to grind it with? You're not going to grind it with water, because the pigments that we are going to talk about are not soluble in water. They are only soluble in organic solvents like alcohol or acetone, those kind of things it is soluble. See here, look, I told you that the pigments are present here. They are lipid soluble. If they are lipid soluble, they will not like water, they will not dissolve in water. So even if you grind your leaf sample with water, it will not give out color, right? So here you can see that the pigments are lipid soluble. So to make it dissolve, you are going to use some solvent like acetone or alcohol. Okay, so you're going to use it and grind it nicely. Then strain it using a filter paper. You're going to get this nice decoction of the leaf. What you're going to do next is, you will take a kind of paper called Whatman paper. It's also called chromatographic paper. What you do there is, at one end of this chromatographic paper or Whatman paper, you will take a capillary tube, small capillary tube. You take a little bit of this leaf sample and then you blot it here. In the center like this, you're going to blot it. Whichever solvent you use to dissolve this pigments, you will take another beaker and fill that beaker only at the bottom with the same solvent. Usually it will be a mixture of alcohol, acetone, all of them. Now you will suspend this into this liquid. Just at the tip, this paper is touching the liquid. You will not immerse this paper in liquid. Just at the tip, it should touch the liquid. So what does this paper do? It will start absorbing that solvent. And at the center, you had added this, right? This blot which contained this extract that you got from the leaf. As the solvent gets absorbed and moves up the chromatographic paper or the Whatman paper, it will separate the pigments out. Whichever pigments are present here, they will all start to get separated. So at the end of this experiment, you can see something like this. Sometimes it appears as bands, sometimes you can see peaks like this. Okay, what is the principle? How does it separate? All that we don't need. Okay, so by doing something called paper chromatography, what I just told you right now, you can see that the pigments will separate. From this, you can get to know that leaves majorly have 4 types of pigments. You have chlorophyll A, which is bright green or blue-green in color, that will separate out. You have chlorophyll B, which is also green, but it is somewhat yellow-green in color. You have xanthophylls, which are yellow in color. Carotenes are yellowish orange in color. You can say orange or yellowish orange. So from this I know, in the leaf, there are more pigments other than chlorophyll A. Chlorophyll A is the primary pigment that is directly involved in photosynthesis, but these guys also kind of help chlorophyll A. Okay, so if you've noticed, when you take a leaf and soak it in water, the leaf does not bleed color, right? The leaf does not let go of the green color and become pale white. If you take a carrot which has carotene, it's orange in color, and place it in water, hot or cold, it will not lose its color and become white. The reason why it's not losing color is because that pigment is present deep inside the lipid membrane. It is not soluble in water at all. Even if you say, sometimes when you make a palak or some soppu sambar, sometimes the samba turns green in color. What happens then? Then you're heating it. When you add hot water to it, the plant wall and the structure of the plant cell will disintegrate such that it will release the chlorophyll outside of the cell. The structure itself is damaged, and if there is some oil or something, then it can dissolve in it and it will show color. Otherwise, if you take just one plant and grind it in water, it will not shed color unless the plant cell itself is destroyed. If you talk about beetroots, just soak a piece of beetroot in a glass of water. It will immediately release color. Take a pomegranate seed, squish it, your fingers will turn pink. What is the difference then? Why are these colors not shedding? Why are those colors shedding? Are they not present in the chloroplast? Are they not present in the membrane? How is that they are getting dissolving in water but these guys aren't? The reason why those purple, blue kind of pigments shed color, like in blueberries also you may have seen, in raspberries you may have seen, it will release color, is because over there the pigments are called anthocyanins. These anthocyanins are not located inside the chloroplast membranes. Instead, they are dissolved in the sap of the vacuole. They are present inside the vacuole. So if it is present inside the vacuole, it means obviously it is water soluble. So when pigments are water soluble, you add water, it will shed color. If pigments are not water soluble, like the pigments that we just saw, if they are lipid soluble, even if you add water, it will not shed color. Imagine if these chlorophyll pigments were water soluble. What would happen after a heavy rain? The entire plant that was green, had green leaves, will shed its color and will become white, right? It will become very pale. So that should not happen. That is the reason why pigments, which are absolutely essential for plants, for their survival, for photosynthesis, they are not water soluble, they are lipid soluble, and they are present inside, deep inside the membranes of the chloroplast. Okay, so these are some of the pigments that you can see in our leaves. Now, I told you chlorophyll A, chlorophyll B. There are two types of chlorophyll in higher plants that we see, but of course in algae you can see chlorophyll C, chlorophyll D, other different types. But in this chapter we are focusing on photosynthesis in higher plants. So we will discuss only chlorophyll A and B, which is found here. If you look at the structure of chlorophyll A, this is what it looks like. It has a porphyrin ring. This is not important. I just want you to see this because in tomorrow's class I will bring this up. In the center, there is a magnesium. Okay, this is the porphyrin head. You can see alternating double and single bonds will be present, not important. And then it will have a very long hydrocarbon tail. It has a lot of carbon. See, this is the structure of chlorophyll. What is the difference in chlorophyll A and chlorophyll B? The difference between chlorophyll A and chlorophyll B is in this R group here. In chlorophyll A, it is CH3. In chlorophyll B, it is CHO. That's the only difference. Otherwise, apart from that, everything else is the same. This small structural difference is what we use to classify chlorophyll A and chlorophyll B. Does this really matter? Why should we classify it? Because what wavelength of light a pigment molecule absorbs depends on its structure. Because of this slight structural difference that you see in chlorophyll, chlorophyll A absorbs a different wavelength of light. It prefers a different wavelength of light, and chlorophyll B prefers a different wavelength of light. Is it clear so far? Does anybody have any doubts? Let me know. Okay. Now, let's learn about something called absorption spectrum. Gnanavi, yes, they are released inside the stroma. Yeah, stromal lamellae are also called fret lamellae. Cool. We will study about something called absorption spectrum. How can you find out what wavelengths of light a particular pigment will prefer? Okay, so does it absorb maximum red light? Yesterday's class, we saw how in T.W. Engelmann's experiment, he used cladophora and aerobic bacteria. He got to know that plants mostly prefer wavelengths in the red region and in the violet region, right? That is one way of finding it out. But if you have to find out specifically at what wavelength the pigment prefers, at what wavelength of light the pigment prefers, you can do something called the absorption spectrum. Over here, what do we call anything a pigment? When you're calling something a pigment, it means it has the ability to absorb some wavelength of light in the visible spectrum and reflect some wavelengths of light and appear colored. Okay. Such molecules are called pigments. Right now we saw that within plant leaves you have 4 kinds of pigments. You have chlorophyll A, chlorophyll B, xanthophylls and carotenes. Okay. So each of these xanthophylls and carotenes together are called carotenoids. Okay. So these pigments, we will learn about how you can find out which wavelength of light it prefers. For that we use an instrument called a spectrophotometer. What we are going to do in this spectrophotometer is, this is the spectrophotometer. So what we do here is, you will separate out the different pigments over here, right? In chromatography, you can separate chlorophyll A, B, xanthophylls, carotenes, right? Similar to how pigments are separated here, there are other techniques by which you can separate individual pigments. Once I've separated individual pigments, I will isolate chlorophyll A alone. I will take chlorophyll A pigment alone, put it in a tube like this, which is called a cuvette. Okay, so I will take just this chlorophyll A in the cuvette and place it inside this instrument called spectrophotometer. Now what happens inside the spectrophotometer? There is provision to incident with chlorophyll specific wavelengths of light. Okay, so I can choose to incident. See, in this within the spectrophotometer, all of this is present. Okay, so this is how the inside of the spectrophotometer will look. There will be a light source that will direct the light towards a prism. This prism will split the light into its individual components. If I want to check in the yellow region how much absorption chlorophyll A shows, there will be a slit over here. That is called the wavelength selector. I can adjust this. I can move this to red. I can move this to, there will be like regulators there. I can move this to red, orange, yellow, green, blue, whatever regions I want. I can select it there. Okay, so I can move this slit around. According to that, I will choose one of these colors. Then, if I am incidenting or illuminating this chlorophyll A with yellow color wavelength, okay, some amount of it will be absorbed by the pigment. Some amount of it will be transmitted back, right? It will not absorb everything that hits it. Some amount will be absorbed, some amount will be transmitted. Now this instrument knows to calculate how much has been absorbed based on how much has been coming out. It knows it put this much was the incident wavelength, this much is coming out. It does some calculation and shows us how much was absorbed. That value is called your absorbance value. Each pigment for each wavelength will have its own characteristic absorbance value. So we do this. What I'm going to do first, I will take chlorophyll A, I will put it inside this cuvette, I will take this cuvette and put it inside that holder that there is. I will close this. I will set it first at violet, 400 nanometer. I will check what is the absorbance. I will make note of it. Then I will move it to 450 nanometer. I will check how much is absorbed, how much is transmitted, what is the absorbance value. I will make note of it. Like that, I will do it for all the wavelengths that I want. So story of chlorophyll A is over. Next, I will take chlorophyll B. I will repeat the same thing for chlorophyll B. Then I will take carotenoids. I will repeat the same thing for carotenoids. I have a set of data. Now I'm going to plot this data in a graph. When I do that, that is called your absorption spectrum. So on an absorption spectrum, in the x-axis, you have various wavelengths of light in the visible spectrum. You have it from 400 to 700 nanometer. Okay, now let's just focus on chlorophyll A, this bright green colored line that I'm drawing. This is what is the absorption spectrum of chlorophyll A. How did I get to this? At 400 nanometer, if the absorbance is this much, I will plot it here. At 450, if absorbance is this much, I will mark it there. At 500, if absorbance is this much, I will mark it there. Likewise, I will mark it everywhere and then I will join the dots. So what I get as a result is the absorption spectrum of chlorophyll A. Likewise, I will do it for chlorophyll B as well. I will mark this in yellow to differentiate the color, but chlorophyll B is also green in color. Okay, so here you can see this is the absorption spectrum for chlorophyll. Despite being 99.9% similar in structure, I want you to see how the absorption spectrum for both is so different. Right? I told you there's only one change between chlorophyll A and chlorophyll B. Despite that small change, you can see how much absorbance is affected in both these molecules, both these pigments. Right? And then we will do something similar for carotenoids as well. So this is the kind of graph that you will get. Okay? So this is what the absorption spectrum is. From this, what will I get to know? I will get to know that which is the wavelength that pigments can absorb most efficiently, maximally. Which is the most preferred wavelength for a pigment, I can identify from this. Alright, now we have something similar called an action spectrum. In action spectrum, we do not do it for individual pigments. We do it for a plant. Now in the action spectrum, we are looking for at what wavelengths photosynthesis is maximum. How do you check for the rate of photosynthesis? If you want to find out at what rate photosynthesis is proceeding, is it happening fast, is it slowed down, at what wavelengths maximum output I will get, at what wavelength the production of glucose and oxygen is very low, I want to find that out. I want to find out at what wavelength photosynthesis happens maximally. How I can do that is by measuring the products of photosynthesis. I will give one wavelength of light to the plant. Illuminate the plant with a monochromatic light like 400 nanometers. That's the only wavelength of light that I'm, illuminating the plant with. So I will check for the photosynthetic output. How many glucose molecules are forming in that light is one way to check it, or how many oxygen molecules are being released, that is another way to check it. If you have to check for glucose molecules that are produced, that is a tedious task, it is difficult. But how many oxygen molecules are being produced, that is relatively easier. So over here I will check for how much oxygen evolution is happening, because oxygen is coming as a product of photosynthesis. If photosynthesis rate is happening fast, then the number of oxygen molecules that are produced also will be high. So output will be high. So if more oxygen is produced, I can say, okay, photosynthesis is happening at a decent rate. If oxygen molecules that are produced is less, then photosynthesis is happening at a slow rate. That is something that I can see. So over here, what we do is we illuminate the plant with various wavelengths of light, and what we are checking here is not absorbance. We are measuring how much oxygen is released. If you're illuminating the plant with just 400 nanometer, let's say, this is how much oxygen is released. If you're illuminating the plant with 450, this is how much oxygen is released. If you do this at 500 nanometer, this is how much oxygen is released. So wherever peaks are there, that shows at that particular wavelength, photosynthetic rate is very high. Okay. Over here you have another peak. We can say that at this wavelength, photosynthetic rate is very high. Okay. So from this we will get to know at what wavelengths photosynthesis rate is very high. Now, if you try to compare this action spectrum with the absorption spectrum, if you compare the action spectrum with absorption spectrum and look at the absorption spectrum of all the three pigments, okay, in the leaf, all 4 types of pigments are present, 3 types of pigments are present. But you want to find out which of them is directly involved in photosynthesis. So you compare the peaks of chlorophyll B, chlorophyll A and carotenoids with the action spectrum. You will notice that wherever there is peak for chlorophyll A, somewhere around the same wavelength there is peak for action spectrum as well. Wherever there is peak, another peak in chlorophyll A absorption spectrum, if not exact, somewhere very close to it, you can see a peak in action spectrum as well. What does this indicate? Compare this with the others. The peak, where the peak is present, that is different in the action spectrum and the absorption spectrum of chlorophyll B and carotenoids. But if you take the absorption spectrum of chlorophyll A and compare it, superpose it on the action spectrum of photosynthesis, you can see that the peaks are somewhat matching. This kind of indicates that chlorophyll A is the major pigment involved. Wherever chlorophyll A is absorbing more wavelength of light, at that point itself, action spectrum is showing a peak. At that point itself, more oxygen molecules are being produced. So this kind of shows us that though the leaf has many types of pigments, chlorophyll A is the primary pigment. It is dependent on chlorophyll, the rate of photosynthesis. Okay. So, yeah, that's about it. Have you understood the difference between action and absorption spectrum? Do you have any doubts here? Just 1 minute, I'll bring some water and come back. Don't create a scene in the chat box. Hey, class is not over. Class is not over. Stay back. Don't run away. By mistake, I put the thank you slide. I'm back. Correct, Kirtana. Oh. Yes, Janani. ATP is released in stroma only, va? Okay. Let's talk about pigments. This reminds me of offline classes. I told you that I'll be back in 1 minute. I'm going to drink water. Everybody's celebrating already. Class over. Class over. Okay, can you all calm down? Can we get back to business? I'll disable the comments. Pigments are basically molecules that absorb specific wavelengths of light. Now we spoke about how many pigments? We spoke of chlorophyll A, which is our most important pigment in photosynthesis. And then we have our second pigment, which is kind of greenish yellow. Okay, this color looks good. Chlorophyll B. Then we have Xanthophylls. Okay, let me just write it carotenoids. Now each of these has a specific wavelength of light that it prefers to absorb. For chlorophyll A, it is about 650 to 700 nanometer. For chlorophyll B, it is around 620 to 650 nanometers. For carotenes, it is around 450 to 500 nanometer. You can see how each one pigment So all of these pigments are present and it's forming like a complex. Okay. I told you, in the thylakoid membranes and the membranes of the chloroplast, you can find hundreds of pigments present as pigment complexes. So over here, all of these pigments are present. Okay. In each pigment complex or photosystem as we call it, or a light harvesting complex we call it. So what happens is you have hundreds of pigments. Of all those pigments, the most important pigment is your chlorophyll A. Chlorophyll A is called the reaction center. This is primarily involved in photosynthesis. Chlorophyll B, carotenes, all of these are not directly involved. Of course, they play a very, very important role, but they are directly not involved in photosynthesis. So these are called antenna pigments or accessory pigments. These are not arranged in any way that it likes. There is a specific order for the arrangement of pigments in a pigment complex. Okay, at the center you will always find chlorophyll A molecule. So in the center you have a molecule of chlorophyll A. Surrounding this chlorophyll A, you will have other pigments such as chlorophyll B, you will have xanthophylls, and you will even have carotenoids. Okay, so in the center there is chlorophyll A. Around that you have chlorophyll B. Here you have Xanthophylls. Here you have Carotenes. Okay, so these pigments are present. If the action spectrum is perfectly correlating or happening according to chlorophyll A, then you might ask, why are the other pigments even present? Why can't leaves just, do with chlorophyll A? What is the need for other pigments if they're not directly involved? I will tell you. This chlorophyll A has the ability to absorb only 650 to 700 nanometers wavelength of light. When sunlight is hitting on it, only 650 to 700 nanometer wavelength of sunlight will not hit. Other wavelengths also will be present. But this chlorophyll A lacks the ability to absorb those wavelengths. So the energy in those wavelengths of light will go wasted. What can happen if these pigments are present instead? Each of these pigments has its own range which it prefers to absorb. So what if chlorophyll A cannot absorb it? These guys will absorb it, use that energy and transfer that energy to chlorophyll. Chlorophyll cannot directly absorb these wavelengths. But what can it do? What these pigments will help chlorophyll in is, they will absorb these wavelengths of light which chlorophyll A cannot. The energy that they get from absorbing lights of different wavelength, they will transfer that energy to chlorophyll A that is present in the center. You may have learnt in physics that when wavelength is high, frequency will be low, the energy also will be low. When energy has to be transferred from one pigment to another, it can always get transferred from where there is more energy to where there is less energy. So what happens here is, which ones have more energy? Which wavelengths of light have more energy? 450 to 500, obviously wavelength is less, energy is more. So that energy is captured by these carotenoids and they will pass it on to chlorophyll B, because chlorophyll B absorbs comparatively lesser energy. Now from chlorophyll B, it will get passed on to chlorophyll A. Though chlorophyll A cannot absorb many wavelengths of light, because of the presence of these guys, chlorophyll A will get the energy of the other wavelengths as well, even though by itself it cannot absorb it. So that is one reason why these pigments play an important role. Another thing is, in photosynthesis you will see there is a lot of electron transfer, transport, all of that happening. A lot of oxidation reactions will be happening. As a result of that, a lot of free radicals will be produced. Free radicals are very damaging to the body. You may have read about that. That's why we should consume antioxidants and all that is there, right? So all of these free radicals will be present that could damage the cell, that could damage the chlorophyll pigment itself. So these carotenoids and the other pigments, accessory pigments that are there, will protect this chlorophyll A from that kind of damaging effects of free radicals. And sometimes when the intensity of light is too high, if these pigments are not there, chlorophyll A alone is there, it can get damaged. Because of the presence of these pigments, even at high light intensities, chlorophyll A gets protected. So these guys are acting like a very good support system, very good friends for chlorophyll A, supporting it in everything and making sure it performs photosynthesis so that the plant as a whole survives. Alright, so this is what is about the pigments, the different kinds of pigments. Now I told you this is a pigment complex. It has many pigments present in it. Now along with these pigments, you will also find certain proteins present. Okay, certain proteins will be present. The pigments plus these proteins, some of which are enzymes and some of the electron carriers, all will be present. All of that combined together is called a photosystem. Photosystem. In the photosystem, you will have reaction center also. In the photosystem, you will have these antennae molecules or accessory pigments as well. What do these antennae molecules do? What does an antenna do? Antenna in your car or antenna in radios we used to get earlier, it captures the signal and passes it on, right? Similarly, here it captures the energy and passes it on to chlorophyll A. So in a photosystem, you have pigments, which includes the reaction center and the accessory pigments. Along with that, some proteins will be present, some enzymes will be present, some electron carriers will be present. All of that together is what we call a photosystem. Okay, so just the reaction center and antenna pigments, just those, you can call them the light harvesting complex. Light harvesting complex. The pigment complex can be called light harvesting complex. Over there, in each light harvesting complex, you will find in the reaction center one molecule of chlorophyll A, and in the antenna molecules, you can find hundreds of chlorophyll B and carotene, xanthophylls, all of that. So this is called a photosystem. In short, we call it PS. Now, where is this photosystem present? This photosystem is present right here in the thylakoid membrane. So the pigment complexes along with proteins, enzymes, electron carriers form photosystems, and they are present in this membrane system. Now, there are two types of photosystems. Okay, we learn about that. There are two types of photosystems involved in photosynthesis light reaction. Photosystem. It is of two types. The first one is called PS1 or photosystem 1. The second one is called PS2 or photosystem 2. What is the difference between these two? Why are we calling this PS1, PS2? Because this was first discovered. PS1 was first discovered, so it's called, PS1 because that was first discovered. PS2 was discovered later. What about the pigments in PS1? The pigments, if you have to talk about the pigments, specifically, let's say the reaction center. In the reaction center here, you have chlorophyll A, obviously. But this chlorophyll A absorbs wavelength of light 700. Okay, so we call this chlorophyll A 700. In PS2, the reaction center will absorb, in the reaction center you have chlorophyll A itself, and that will absorb a wavelength of light 680 nanometers. So we call it chlorophyll A 680. Now, where is PS1 located? PS1 is located in the grana lamellae or orala and stroma lamellae. PS2 is located in the grana lamellae only. For PS1 to function, it requires wavelength of light that is less than 700 nanometer. 700 nanometer is maximum wavelength that it can absorb. If it is beyond 700 nanometer, then the energy in that will be less. right? So it will absorb wavelengths less than 700 nanometer, which will have more energy. This one will function only when it is incident with lights that have wavelength less than 680 nanometers. Understood? These are the differences between photosystem 1 and photosystem 2. If you think this is the chloroplast inner membrane, this is the thylakoid membrane, these are the stroma lamellae. You can find pigment complexes here, here as well. So this PS1 is present in the grana lamellae. It's also present in the stroma lamellae. This photosystem 2 is present only in the grana lamellae. Is it clear? Okay, so quickly answer some questions before we end the session. Just 1 minute. You know? What is the correct answer? Site of light reaction. Dark reaction. PS1 site. Reaction center of PS1, chlorophyll A, 700, chlorophyll A, 680. One more thing, for the post topic, there are no questions that I could find PYQs in Neet, which was asked from what was taught today. Doesn't mean it will not be asked. You can get questions from anything and anywhere and everywhere. So be prepared anyway. I'm going very slow in this chapter because I want you all to understand, like it and learn it. Most people hate plant physiology. I want to change that for you. Nicotinamide, adenine, dinucleotide, phosphate. Yeah, you can say grana and stroma lamellae also, no problem. We are doing just little bit in this chapter, little we're going very slow. Okay, please read today itself. If you have any doubts, we can discuss that tomorrow. Sanjana, it's nicotinamide adenine dinucleotide phosphate, not adenosine. All right, thank you. Tomorrow's class, we will learn about light reaction very slowly. Okay? I'll see you tomorrow. In the meantime, if you want to share your issues and have your concerns addressed, please feel free to fill the form. Okay? Thank you so much. Bye-bye. See you tomorrow sharp at 12 o'clock. Bye. For this coming test, I have not included this chapter. You will only have questions from anatomy. Okay?